Hi, my name is Ken Lasseson. I wrote the Microbiome Prescription website. I am a high-functioning autistic person. If you listen to my speech, you hear something which sounds like an accent. I was born in the Pacific Northwest. Parents were immigrants, but they did not have accents. They basically, actually, yes, immigrants coming over to Canada when they were 3 years old. Technically, they're immigrants, but basically zero speech. So you can hear in my speech the consequence of not learning to speak until I was 9 years old. Both my parents were in their 40s when I was born, so very much a classic background for somebody with autism from, should I say, 7 years ago when it was unknown. I survived, I adapted, and as many autistic people I became very good in mathematics and in tunnel vision or monofocus and things, which was successful in my case because of some good advice by my teachers along the way of what I should go into and what I should avoid. Okay, so much for me. Let's go over and let's talk about autism microbiome. And my purpose of the talk is to do a walkthrough about reducing symptoms by microbiome manipulation. Okay, so you may want to say, okay, isn't microbiome DNA, isn't it a ton of other things? Isn't because of vaccine? Well, the thing is, we find that when you go over to look at PubMed studies, you'll find almost all cognitive issues, including Alzheimer's and Parkinson's, have abnormalities reported in the microbiome. And there have been some studies showing that if you modify the microbiome, the severity of the symptoms decrease. So down here you see things, for example, for akkermansia, you will see high akkermansia is pretty much across the board for Parkinson's disease. Um, Other things you see some reporting high, some reporting low. So there is a problem with consistency, and at this point of time, we end up getting into some rather nasty realities. We have many, many studies reporting shifts, but the studies do not agree with each other. Sometimes they cannot be replicated. In few cases, like for Parkinson, you get a massive amount of agreement, and for other things. But often, some things say some high, some things say low. The reason could be some cases because we're dealing with different subclasses of autism or Parkinson's or Alzheimer's. And hence you get, depending on how you select the participants, you got things going one way or the other way with a different subset. So there are many things to deal with it. And it adds to the complexity. But let's not quite go on to that problem selection. Let's go on to something which to me is much more important, which is that all these were done by using different ways of getting the names and quantities from the microbiome. I talked to Scott Jackson, the National Institute of Science and Technology lead for microbiome system, and basically right now you take the same sample and you get, well, if this was from 2019, you get more than 97 different answers. 97 different answers from a study. And these are different answers not from which lab equipment is used. This is different answer when you put a sample through a processor, which now produces a data file called a FASTQ file. And now this one has to be interpreted by software. Keyword software. The catch is there's no standardization for software. The result is you get 97 different answers, and I've seen that from my own sample. I was using uBiome until I went bankrupt, and then for a while there, I was able to upload the data to 4 other providers who took the same physical file and processed it, and I had massive disagreement all over the place as to what was in the sample. Same data file, identical same data file, They processed it with different software, different reference library, and they came up with different things, which means we have an immediate problem. You cannot mix results from labs. The problem with studies is they're done with all sorts of different labs. So we have a site challenge here. So let's go over to microbiome prescription. Some stats there. Um, the problems we have is different lab processing. Often studies are done on small sample sizes, and often we have naive statistical methods. Naive as in a sense, unlike me who has a master's in statistics and some PhD courses in the area, they use the standard textbook, calculate the average, calculate the mean, apply just formula, cookbook formulas, and the result is they miss a huge quantity of data. Okay, so when we go over to microbiome prescription, we find we have had people upload 4,500 different samples. Instead of dealing with 30 samples, we have 4,500. We have 263 of these are annotated as coming from people with autism. For that, if you break down by lab and by condition, we have 103 biome sites samples marked with autism and 93 ombre sites with autism. Those are the two top ones there. And what we're going to do is we're going to go and walk through using the site how if you have a kid with autism or you have autism yourself, you can use the site to probably moderate or improve the symptoms. So, quick summary. We're going to be using our own association of bacteria to autism using the same lab and everything. So we eliminate the first major problem, which is non-reproducibility of results. We have the same lab being used, and we have people self-declaring that the person has autism. We could then work using conventional literature alone, which has some problems with it, but it's the best we can do. Then we're going to look at a novel probiotic suggestion, which is what I'm personally excited for. I've actually tried for myself, and it has worked. It has worked surprisingly well. And then we're going to look, if we are lucky, look at cross-validated suggestions for autism. That tends to depend on how much published research is there. But I'm hoping at this point in time, before going into it, that we do it. If not, sometime in the future, we hopefully will be there. Okay, so let's go over to the site. Okay, so I've grabbed a sample of somebody who reported autism, went to the site, logged in, and the first thing it comes up with just novice. Just tell me what to do. You really, it's because you have a diagnosis and perhaps one or more symptoms, you want to actually go down to the next one, beginner symptoms, which in a few seconds will pop up, and voila, we see that we have autism person sitting at the 88th percentile, so he is a good match. He marked it as autism, and what you do end up doing is you're going through the list, and it's based on the prediction of what somebody may have, based on the bacteria should see, and sometimes it may be correct, sometimes it won't. But in general, the main advantage is that they allow us to identify which bacteria are likely associated. So you can go down list and mark off whichever one you have. And some, for example, this one we lucky autism on the top. Sometimes it may be a bit further down, but 88 percentile, so it is a reasonably good match. And all we now do is go over here and we look at the type of suggestions which we have here, and it defaults to check everything, and we basically do nothing more complicated than do a get suggestions. And it now lists at the top what symptoms you have annotated, and now it goes and lists the bacteria and what the nature of it, which you find there are 16 bacteria where the pattern matches up above. And then we have down below, we have suggestions of things to take. And they're a bit all over the place because they're based on what is available in the literature. So we have things there. We have some things antibiotics. We have other things here. We have also have things to avoid, and sometimes it can be quite interesting. For example, take vitamin B3, avoid vitamin B2, which means a complex B vitamin supplement probably should not be taken. You want to be as expensive. Again, these are things calculated individually to help make the adjustment of these bacteria. Not guaranteed you, they just have a higher probability. Remember, I'm training stats. I work on probabilities, and these are things that are likely to make the shifts worse. So we have everything there now. In this case, no probiotics can be found, which suggests using R2, which we'll come to shortly. But also, if you go up to there, we have consensus report, which goes and takes a look at data. Now, to get this data, what we do is we run the data through 4 to 5 different algorithms and come up with the most common suggestion. If you want to see the actual detail, just go over here. And there you see counts. For example, all 5 algorithms says take vitamin B3 and go there now. You may be interested in one thing alone, like for example herbs or spices. And thereby we have here are the herbs which are likely to help and the relative weight. And we also have, if you click that, sorry, in reverse, we have things which are unlikely to help. And again, everything is based on the literature, and sometimes you can get things like marijuana here is not likely to help, but the cannabinoids from marijuana, which is in marijuana, is likely to help. And often what we have is studies look at either a fine component or a broad substance, and sometimes the broad substance help, but the component doesn't. Sometimes the component help, but the broad substance. And the reason is the broad substance is loaded with other chemicals, and those other chemicals have additional effects. So you will find this sort of flip, oh, wait a minute, this doesn't agree with that. It does if you keep in mind that we're talking with one part or many parts. Then you can realize, wait a minute, okay, this one part of marijuana helps. The rest of marijuana doesn't help. So there we have there, and in general, what I suggest is to look at the highest value. That's 720 here, which means that the first choice item would be anything which is half that value or more. So you have things here. You have things like antivirals. You have whole grain wheats, refined wheat bread are all there. Um, and you can go down and then take a look. So you get an idea. The ideal thing is always ones which are nothing but to take and absolutely the super ideal will be no avoids. Then you are pretty safe as doing something which has the highest probability. If it's a little there, then okay, then yeah, probably worth taking it. Okay, so that is the cross-validation, and you can sort by whatever you want there. Now let's go back to the prior page. We also had the food planner menu, which I will flip over to very quickly. And what it does, it identifies the nutrients which you want to increase or you want to decrease. Now, one to catch is that most of the time, like this chemical here, you have absolutely no idea what food it is. The food database looks at a whole bunch of publicly accessible data, food databases and produce there, and you find, oh, okay, that thing, this single thing happens to be in red currant, raw red currant jam, which is sweet, okay. But we can go back to the there, or we can go and say, okay, let's take a look at quick avoid foods. As you see, the list comes from around the world, but we see that Turkey really dominates as a to avoid. And if you go back to quick take foods, we see kiwi, wheat, red currant, turmeric, apricot, jam. We find all sorts of food which probably should be made a regular part of the diet. And sometimes you get some interesting things like, wait a minute, sponge cake? Well, the kid would be happy. Let's see why. Click here. And the reason is it has two things in it, glucose and carbohydrates. Those are the two things which are good in it, which are dominant. We also have fructose, and the negative thing is polyunsaturated. So that is a very quick introduction to making use of the there. Okay, so that gives you good idea of it. Notice the key thing: when in doubt, leave it out. If there is what appears to be mutually contradictory suggestions, often because of the one component big mixture thing, if in doubt, leave it out. This is not a protocol. It is giving individual suggestions. You can cherry pick them to your heart's content. Every item is estimated to help improve things. For the majority of people, you may or may not be one of the lucky ones, or actually you're probably more unlucky because it should work for most people. Okay, so now we end up with the last thing, which is the taking these things. It will go over and do some fancy calculations and come up with the probiotic species there. And some of these are in experiments in research right now. Hopefully they come on the market. At the moment they're not. Endocarpus feliceum is on the market, and you see the good aspect and the bad aspect. Again, generally I take good minus bad and you get a net number. The idea would be just good and no bad, in which case we have Lactococcus latus, and we have been on a typical which is available from one seller is claiming to have it there. And then we have number one, which is a probiotic, but probably going to be real hard to find. Lactococcus is also easily available probiotic. Number one here, which I think you can get. I think I have some in my fridge. So you have to go through there and find out which ones are there. But the ideal thing is all good, no bad. But if it's a mixture, then you may want to try doing it. If you can get Anaconda feliceum, I've used personally and I found quite effective. And that one I also have taken and it is effective. And Akkermansia municipa, another one. In fact, my favorite is Akkermansia municipa, Lactococcus cremelius, and Endococcus feliceum. That's combination I've used, and in fact just finished a 2-week cycle of it, and being very, very happy with the results. A little bit of constipation, but not too bad. Which comes back to the issue. Probiotics, and for herbs, you want to do on a fortnight, every 2 weeks, change them constantly. And the reason is the simple parable is that the bad bacteria will adapt to them, and the mutations to resistance will suddenly keep growing, and you end up with the same quantity of bad bacteria a few months later, although you're taking the same thing because they are keyword. The equivalent of antibiotic resistance or probiotic resistance or herb resistance, all those terms apply. What doesn't get killed off, which usually is a small percentage, maybe 1% or 5%, will just keep regrowing. And if you keep giving it, hey, we have resisted, we're going to grow bigger each time. So you want to rotate herbs, probiotics, antibiotics, maximum of 2 weeks on any one, and rotate it. That's my philosophy. I got that from Dr. Cecile Zardine, who is treating chronic fatigue syndrome in South Africa for 20 years. And that came from actually her father's experience working for the Pasteur Institute for Tropical Medicine, dealing with chronic rickettsia. and they discovered that the secret was the rotation, not giving more or higher dosages, it was rotation. Okay, now I'm going to go over to my last way of doing things. I'm hoping it's there. So I'm going to go back to the home page. I'm going to go to old UI. Actually, I think I have a direct. Actually, I'm going to go back because I think I have a direct link on the page. Let's go back. Uh, okay, yep. Right here, bottom one there takes you direct, saves you some time. And if you go down here, right there you have the things and autism. 66% match for existing studies. Now, I'm happy because this will give you a document which would be convincing to a medical professional of what you should be taking. You go down here and you get reports, and you need to wait a little while because believe me, it does a ton of calculations. Fortunately, I have coffee handy. And we wait a little bit. Ah, there we go. So, up at the top gives you some canned information, which comes from one of the large language models, which is top popular AI. It's basically just padding there. Now it goes identifies all the bacteria shifts that matches what this person has. And notice after everything, it gives you numbers. This is how many of the clinical studies have reported this shift for autism. So in other words, doesn't hey, that is there. Ackerman low, remember we saw that? We have 4 studies reporting that. For Akkermansia the genus, we have 5 studies reporting that. Um, so we now have a match up to what the medical literature has reported are the bacteria shift for autism. And we have the link to the study, so we can just click on here and we'll go and we'll see the study involved. And there we go, and they're at the top. So let's go and let's go to the end. And we have 122 there. Actually, there's more than 122 studies used, but I just give a sampling because I don't like being too tough. And now we have the cross-validated suggestions. These are things from literature which are reported as helping autism. Now, you see some have a good sac. Lactobacillus plantarum is really sitting there nicely. So we now we have vitamin B12, uh, there, CoQ10, kefir, vitamin D. So this is what the literature report helps autism, and these are the same substances which we calculate will help the bacteria shifts up here. So, in other words, we have it cross-validated. These substances will help autism for independent studies, may not deal with the microbiome at all, just somebody tried and help some aspect of autism. And up here we have the bacteria and other studies showing that these substances shift these bacteria in the right direction. And this is why it's called double validated. It is a sweet thing. And now down below we go and have from the R2 suggestions, we have things listed there. And hey, right there, plantarum is sitting there. The sweet thing is, if something is on the cross-validated list and on the R2 thing, that is, I would say, triple validated. You have the suggestion really, really coming out strongly as something that should help. And then down below, we give the alternative names because tomato, boot of a car versus the trunk of a car, the bonnet versus the hood of a car. Different parts of the world call the same thing different names. So here it's basically trying to find the names that you could be using. And now below we have bacteria by bacteria, this bacteria, which is going to be impacted that they are. And here's the thing. Wait a minute, 255, there's only, or 3,055. 3,055, 3162, what? There's only 112 at the bottom. Click here, and you go over to the study where the information is coming from. So, we end up having complete documentation so you can trace back to every little bit of information. And we're talking about 3,200 reference studies are used to produce this page. That is a lot of reading, believe me, I know it. One aspect of having autism is this super focus and not getting bored by doing things that are somewhat boring and mundane. But there you go, and there we have all the things there. And below that, we have, it's a long report. If you print out to your MD, he's probably going to ask, how many trees did it cost? And now we have additional suggestions, which are things which are not up there, but which are computed to help. Often they're not up there because nobody has bothered studying this particular combination of stuff with autism. In other words, it's called sparse data or absence of data. So these are things calculated theoretically to help, hasn't been tried in clinical study, but there are reasonable grounds that they will help. The voids are a real joy because in general, the voids are based on, give me a second to rephrase. Okay, items to avoid will almost never be in a published study. Why? Because people want success. So if substance doesn't work, the study never gets published, or even if it gets submitted somewhere, doesn't get published because it's not a positive result. So here we have items to avoid, which is done based on the model we are using. Now you will find a couple of interesting things happening. Notice wheat is here, but wait a moment, up at the top, when we did it using the bacteria identified from Microbiome Prescription analysis, wheat was a to take. Here it's to an avoid. Why? Is it a wheat on one and take on the other? Because of which bacteria we are focusing on. We don't know which ones they are. Here we are working on what the literature report and where we have a match. Above we are working from the statistics I did on the samples which are being submitted and finding patterns there. Keeps coming back to the D by key model. When in doubt, leave it out. Simple there. Why flip a coin where you're not sure which way it's going to come up, when you can just simply omit something totally and keep the things which you have agreement on. So when in doubt, leave it out. And now last part of the report is the reference and it's a partial list. And just click here to go to the study. So it's nice, simple, well documented, and there. So to recap, what you want to do, go back here, is log in, beginner symptoms, mark all the symptoms person has, or all the major symptoms person has, and then it will do two things. One, it will save it and will add to the database, which means it will help everybody else get better results because we get better identification of bacteria involved. So you are making a contribution that way, and then basically go over. Usually I suggest keeping everything, get the list, then you have the joys of your suggestions, you have the ability to go over and look at the consensus report, go to food menu, which often will find things which are never being studied. What we're working from is what the nutrients which are in, which have been identified to help, working from that point. So it is not as precise as we like, but it is a pretty good, reasonable thing consensus report, do the cross-data report, and you should have two things. One, a big stack of paper on your desk to start reading through and cross-referencing, because remember, we have three different ways of getting things like probiotics. And that is from microbiome prescription, statistical regression, finding which bacteria, or two, which actually is done two times, one on the bacteria which were identified by microbiome prescription, and then we go over to cross-validate it, which uses published studies only, and selected bacteria where you have a match of published studies, and also R2. So we end up actually with four sets of probiotics, which is exception. You can have four different sets of probiotics suggested. Find out what is in common across all of them, and those are the ones with absolutely the best odds of being successful. Okay, at this point in time, I'll simply say that should be it. You have half an hour of data to consume. I've tried getting to the fundamentals. Remember, it is a complicated, evolving area. It gets complicated real fast. But at this point in time, I'm hoping this helps you and helps your child. Bye bye.